Okay, welcome to this explanation of the light-dependent reactions and the Calvin cycle of photosynthesis. Both of these are key steps in the two big key steps in photosynthesis, and they take place within the chloroplast of plant cells. Just to remind you the structure of chloroplasts, I'm just going to show you a kind of side view of one here. Remember, they have a double membrane on the outside and these stacks of thylakoids called grana. So a stack is called grana, a granum, that's just one. And each one of these little bubbles is a thylakoid. I'm going to draw one of those large here so that you can see it. The thylakoids have special protein complexes built into the membrane, and we're going to talk about their function in just a moment. I'm just going to draw one other one which has got a slightly unusual shape down the base here. So the thylakoids are surrounded by a phospholipid bilayer. Goes all the way around them, and in the middle they have this space full of liquid. So here's my phospholipid bilayer. This particular thylakoid seems to have ended up with a rather unusual shape, but that's okay. Okay, so there's my thylakoid, and let me explain. The thylakoid is the site of the light-dependent reactions. I strongly suggest that if you haven't done so already, you pause the video, draw this yourself, and then fill it in as I explain what is happening at each step. So, the thylakoid. It's a bubble, it's got this space in the middle, it's got a phospholipid bilayer around it, which has got special proteins embedded in it. A photon of light hits one of these protein complexes. And these particular protein complexes, this one, is made up of a large number of molecules of a very special protein called chlorophyll. Chlorophyll is a pigment. It gives plants their green color. And the chlorophyll molecules absorb the photon of light. This causes them to excite electrons that are present in the complex. Those electrons get excited, and as a result of this, are passed down a chain of proteins in the membrane. They bounce along, and as they do so, they lose energy. That energy is used to pump hydrogen ions into the thylakoid, like this. This means that there is an increase in the concentration of hydrogen ions inside the thylakoid space, like so. The excitation of electrons actually happens twice in the chain. There is another point in the chain where electrons are again excited, given a high energy state, and so move on. And finally get to this protein at the end of the chain. So, electrons are gaining energy from photons of light using the molecule chlorophyll, and then moving down a protein chain which is used to pump hydrogen ions into the thylakoid. At the end of the chain, the electrons are here. We'll explain what happens to them in a moment. Obviously, as electrons move away from here, we're going to need to replace them. We replace them using electrons from water. Water is split, donating its electrons and producing hydrogen ions, which help to increase the concentration of hydrogen ions in the thylakoid, and oxygen, which will leave the thylakoid and be used for either cellular respiration in the plant or maybe released from the plant, which is very useful for us because, of course, that is the oxygen that we use to breathe. Now, we have an increase in the concentration of hydrogen ions inside the thylakoid. As a result, they're going to want to move out from a high concentration to a low concentration by facilitated diffusion. They do that through this molecule here. This molecule is called ATP synthase. It's a very special enzyme. We know that it's an enzyme because it's got the letters ASE on the end. And ATP synthase uses the energy from the hydrogen ions moving across the membrane to convert ADP, adenosine diphosphate, and inorganic phosphate ions into ATP, which the cell needs for energy. Now, I said we'd come back to it. Back over here, we need to do something with these high energy electrons. They've got a large amount of energy, and the plant cell is going to want to use them. To do this, we use an energy carrier called NADP+. That comes along and it picks up electrons, and it also picks up a hydrogen ion. So we get NADPH formed. This high energy electron carrier is going to be used in our next chemical reaction. So I'm going to draw a dotted line showing it going all the way down to the Calvin cycle. The ATP is also going to be needed to be used in the Calvin cycle. So let's talk about the Calvin cycle. This takes place in the stroma. If we look back to our my little picture that I did at the side of a chloroplast here, the stroma is this section here. It's like the cytoplasm of a chloroplast. The grana was these stacks. This is the stroma. The Calvin cycle is all about producing glucose. If you understand that, then you've got most of the information you need. It's a cycle of reactions that goes round and round. And so you need to draw a circle looking something like that. Now there's lots and lots of enzymes and steps in this reaction. You really don't need to know much of the detail apart from the fact that ATP is being used and NADPH is being used. These two provide the Calvin cycle with energy so that it can turn. There's one other very important molecule that we need to understand here, and it's carbon dioxide. As you know, plants take in carbon dioxide when they photosynthesize to make sugar. And here it is. Here's the carbon dioxide coming into the cycle. We need 6 molecules of carbon dioxide to make 1 glucose molecule. Glucose is also produced by the cycle, so we've got our NAD+, ADP, and glucose is going to be made. Remember that glucose's formula is C6H12O6. So it has 6 carbons in it. That's why we need 6 carbons coming in from carbon dioxide. This is called the Calvin cycle. It's also sometimes called the light-independent reactions. Or the dark reactions. Because it doesn't directly need sunlight. It needs these products, ATP and NADPH, that came from the light-dependent reactions, but this is independent of light. It doesn't need light directly to make it work. There is one enzyme that you need to know that takes place in this cycle. That is because I think you need to know it because it is the most common protein in the world. It's an enzyme called Rubisco. Remember, enzymes only catalyze one chemical reaction. They only catalyze one reaction. But they do it many, many times. Rubisco is quite slow as an enzyme though. So plants have to have a very large amount of Rubisco. Where I've placed it in the cycle gives you a clue as to what its job is. Rubisco grabs hold of carbon dioxide and pulls it into the cycle of chemical reactions. It is the job of Rubisco to bind that unreactive carbon dioxide so that we can transform it into glucose. Without Rubisco, there would be no glucose. Without glucose, we wouldn't be able to, plants wouldn't be able to store their energy, and we wouldn't be able to get food from plants. So that's very useful for us. Remember, this is called the Calvin cycle, and it takes place in the stroma. So, this glucose, what can it do now? Well, it can head off to the cell. It might be transformed into another molecule. It might be used as a source of energy. Let's remember, our balanced chemical equation for photosynthesis is 6H2O that was being used here, plus 6 carbon dioxide, that's from right here, produce 6 molecules of oxygen and our glucose molecule, C6H12O6. That's the balanced equation for photosynthesis. Remember, respiration is just the other way around. Hope you enjoyed this. Study hard.